Okay, so this is a single cell dividing into two. And then after it divides into two, each of those two are going to divide again into four. Four are going to divide into eight, eight into 16. We're just going to keep dividing. So here we have a little lump of cells. This is a little baby animal. And this baby animal will ultimately become a larva, and that larva will undergo metamorphosis and become the sea urchin. So this is sort of early life of a sea urchin, and this is showing the adult state. Okay. So we're going to go through this chapter. We're going to talk about how cells divide, kind of give us an overview of like why cells divide and what the grand scheme of process is. We're going to talk about the cell cycle and how the cell cycle is controlled. And then we're going to talk about prokaryotic cell division. There's a section in here on cancer in the cell cycle. You are welcome to read that. It is interesting. I'm not going to go into that in class. I'm not going to test you on how the cell cycle goes awry when cells become cancerous. Okay, but you're welcome to read that for your own education. Okay, so I have some background material that I think it's important to have to set the context for this chapter. And so I like to do that before we talk about this chapter. Much of this isn't in your book. This is my thing. I might ask you about this stuff, but mostly I want to set the stage and make sure we're all on the same page. And we have a feeling for how and why cell division occurs. So if we think back, we only have a handful of theories in biology, but one of them that we mentioned at the beginning of chapter 4 is the cell theory, right? And we said all cells come from pre-existing cells, right? So this is the way we get new cells. We do cell division so that we can produce new cells. And we might need to produce new cells for a number of different reasons, okay? These cells over here in orange, this is a type of yeast. And yeast are single-celled organisms. So when they reproduce, they're doubling their population. Every time one cell reproduces and becomes two, now we've doubled the number of individuals we have, right? So when they're reproducing, they are just cloning themselves, making a new cell, and growing the population. Okay, so for single-celled organisms, cell division might be reproduction. This tissue that we have here is actually a root, and so you can sort of see this is where the vascular part of the root is going up. And then we have a section here called the meristem, and that's going to be an area of really active division where lots of cells are dividing. And as those cells divide, they push the root down through the soil. So this root tip will extend down further and further, and the root will push through the soil. So that cell division is growth. As those cells divide, the root tip gets longer. Okay, that's for growth. Over here, this gecko has very recently lost its tail, and it is in the process of growing back a new tail, and that new tail is pale in color. It hasn't finished turning on its color patterning to match the original tail, right? Geckos have the ability to lose and regrow their tail as an anti-predator defense. If a cat grabs them by the tail, they can let their tail go and run away and leave the tail wriggling to keep the cat busy while they make their escape. There is some control, but I bet that the control is driven by the amount of force applied. Most of the animals that can do this, like jettison their tail, they actually, like the cells absciss and fracture right between two vertebrae and let go. And so there's going to be a physiological response to a certain amount of pressure exerted or pull occurring or something that causes the cells to absciss and break. It's a very clean break. It's not like we just remember, we're not degloving the tail and leaving the core behind or anything. It comes right off. So regeneration is another purpose of cell division. We're not going to talk about the cell division that humans use for reproduction in this chapter, but we do use cell division for growth. Anytime we get bigger from our original single-celled organism all the way up to our adult size, we're building new cells for growth. We also do regeneration. We can't regenerate our limbs, but if we scrape away some of our tissue, say you crash your bike and you rip open your knee, right? As that grows back, we're regenerating those missing cells and filling in that space. Okay, so repair, growth, regeneration, So I sort of think of repair and regeneration as being together. Did you have a question? Okay. You usually have a question. So in unicellular organisms, cell division is primarily used for reproduction. Going to make another copy of ourselves, grow the population. In multicellular organisms, we can see reproduction. Many multicellular organisms can reproduce just via mitosis, which is the process we're going to talk about in this chapter. Others use a specialized type of cell division for reproduction. And then we also do growth and repair. So instead of regeneration, I call it repair when we think about just maintaining regular body cells. Some of you guys are still writing this. Ready to move on? All right. So when one cell increases its volume and duplicates its genetic material, it becomes sort of one large ungainly cell. And once the two nucleuses have been, the nucleus has been reproduced and completely separated, then that one large ungainly cell will stretch and stretch and stretch until we finally break them apart and have two separate cells. Right? When this happens, we produce two daughter cells. And this is just, again, historically how things were named. We frequently refer to cells as offspring cells, daughter cells. Later, we're going to talk about filial generations. Filial means sons. Some of this is just historic. So the cells that are reproduced are two daughter cells. There's no gender or sex identification to those cells, but we call them daughter cells. Right, so the original cell that you started with is the parent cell, but the two cells that you end up with are both new cells, and that's because both of them contain some of the old stuff, and both of them contain some of the new stuff. And so it's not like all the old stuff stays on one side and all the new stuff goes to the other side. It's actually that, like, as the cell builds more materials, and it just sort of stretches, some old stuff and new stuff goes to this side, and some old stuff and new stuff goes to this side. So they're both kind of new cells, like they have a new composition at the end of it. So we say that they're two new daughter cells. And at the end of cell division, when we produce these cells are identical to each other. So the genetic material is copied exactly the same, so that the contents of the two daughter cells' nucleuses are the same as the original parent cell nucleus. They're identical to the parent cell, and they have a complete set of genetic information. Okay? We will later come back and talk about things like mutations and times when this process goes wrong, and we could have an exception to the identical to the parent cell rule. But by and large, if cell division proceeds as planned and goes correctly, we'll have two identical cells left over. Okay. We also need to make the delineation between asexual reproduction and sexual reproduction. So asexual, that A in front of it means without sex. Right? So asexual reproduction is reproduction without sex. In eukaryotic cells, this is just the process we're going to talk about in this chapter known as mitosis. And you start with a single parent. And that single parent is going to produce a clone of themselves. So the offspring is going to be exactly identical to the parent. In our single-celled yeast over here, parent cell yeast, offspring cell yeast, right? They're dividing a little bit unequally because the one is larger. As that separates, we just have this parent is making an identical copy to itself. Those two cells are going to be the same at the end. Over here, we have starfish. If you cut off the arm of a starfish and you get enough of the central ring, part of the ring canal of the starfish, then that starfish can regenerate a whole new arm. Okay. The arm itself can regenerate a whole new starfish, right? So that is a form of reproduction. There was a time period in Atlantic fishing history in which mussel fishermen were very frustrated with starfish because they felt like starfish were eating too much of their mussel haul. And they decided to kill starfish. So every time they caught a starfish in their net, they would chop it into pieces with their knife and throw it back in the ocean. And I think that is very karmically sound, right? Because all they were doing was causing the starfish to rapidly explode their population as they grew back their cut up limbs. Well, you have to get them into small enough pieces. You have to destroy the central part of the star or desiccate them. Yeah. So there's an invasive starfish that is wreaking havoc on coral reefs, which are already in really bad shape. It's called a crown of thorns. And if you're on a diving trip, usually the person giving you the guide will ask if you want a crown of thorns knife, and you root out the center if you want to kill it, instead of just cutting off the limbs. Not that we should be killing starfish, but invasive species, it's a fine line. Okay then. Compare asexual reproduction to sexual reproduction. In sexual reproduction, we have two parents, right? One of those parents is going to produce one kind of specialized gamete, and the other parent is going to produce a different kind of specialized gametes. So gametes are specialized cells that are produced just for reproduction. Their only purpose is reproduction. They don't do any other task. And they are produced via a separate process called meiosis within specialized reproductive organs. So in order to do sexual reproduction, we have to have a species that has specialized reproductive organs, and those organs have to make unique cells whose sole purpose is to reproduce. And those cells are called gametes. The gametes in animals are referred to as egg and sperm. In plants, we refer to those as egg or ovum and pollen. But we have these two different types of gametes, and those gametes have to fuse in order for reproduction to occur. When this happens, the offspring are genetically unique. And the reason is that when the specialized cells are made, when the egg and sperm are made, the parent is only going to put half of their total genetic information into that cell. So any given egg cell or any given sperm cell has half of their parent's genetic material, not all of it. So that half is going to then recombine with the other parent's half of their genetic material. And even if you have the same two parents reproducing over periods of time, each individual is going to continue to make unique gametes because they're recombining which parts of their genetic material are going into the gamete each time. Okay, so we produce really genetically unique individuals when we carry out sexual reproduction. Many organisms in the animal world can do either asexual reproduction or sexual reproduction. Asexual reproduction is fine when times are good, food's plenty, and the conditions are fine, and we can just chug along. But sexual reproduction is really essential if we might need to be able to carry out natural selection to some changing force, right? Because we get new genetic material, new recombination, new phenotypes, new ways of living when we have sexual reproduction. Sexual reproduction is strongly favored evolutionarily because it gives greater diversity. Ovum, O-V-U-M. Ova or ovum, which is a technical word for egg. So this family up here, I like to show this family because this family is a genetically related family. So these two individuals here and here are essentially grandma and grandpa. And they produced a son. And that son married this woman and produced those 4 grandchildren. Okay. And I pulled this picture from a genetics textbook because I think that we all feel like parents and offspring should look very, very much alike. And in many cases, they do. If you look at pictures of my mom and I all throughout our childhood, if you made my pictures black and white, you couldn't tell which one was which, right? Until about high school, we started to look different. But in many families, that recombining of genetic material leads to offspring that have really diverse appearances. So we have diversity in skin color, diversity in eye color in these offspring because of the recombination of their parents. Okay. There's a lot of potential outcomes, and it's going to depend on which genes join up with which genes when those gametes fuse, and it's also going to depend on environmental conditions. Okay. I also think it's important that we all know how sex happens. I think most of us have the broad picture, but I want to get down to the cellular level. Okay. So this is a human egg, and this is a human sperm with his little happy tail. And inside the egg, we have a nucleus with DNA. We have all the organelles. We've got our cytoplasm full of stuff, organic materials, ions, salts, all of our normal dissolved stuff of the cell. And then we have nutrition. And even in a human, a mammal, where we're going to ultimately feed the developing offspring from the parent's bloodstream, for the first couple weeks, that egg is on its own. And it's going to have to do all of its growth and division of cells based on the nutrition that's housed within that egg. Okay, so there's lots of nutrition there waiting to help produce those new cells. There's also a really big volume, right? Compared to the egg, the sperm is small, right? The sperm is basically DNA, a few mitochondria, and snacks. So basically, the sperm just needs to swim far enough to reach the egg. So it's got to have energy to make that trip, but it can't be too heavy because it's got a long swim upstream, right? So it doesn't have all the organelles. It doesn't have a huge cytoplasmic volume. It's not going to contribute to the nutrient of the egg. It's going to have enough snacks to power its swim. It doesn't want to run out of steam before it gets there. And it's going to have this big long flagella to help it move to its destination. Okay. When this sperm successfully joins with this egg, the nucleus inside the sperm is going to fuse with the nucleus inside the egg and become one nucleus. So technically, this has half the DNA needed to make a functioning offspring. This has half the DNA needed to make a functioning offspring. And it's only when they fuse together that they become a fully formed full-size cell. So when the egg and the sperm come together, even though the egg was one sperm cell and the sperm was one cell, they fuse together to make a single cell. Okay. So meiosis produces these unique gametes, the egg and the sperm. Those egg and sperm fuse together and make that original zygote. And then that fertilized cell just starts dividing. One cell becomes 2 and 4 and 8 and 16 and 32 and 64, right? And they're just going to divide up this egg space. So in the beginning, we're going to just be dividing it up so that this one big cell becomes many small cells. And then the sphere of cells, the lump of cells, is going to start to grow. So over here we have a little baby, our little lump of cells, right? And each one of those cells has been produced by mitosis. Okay? So this cell division process, once we have the fusion of a unique egg and sperm, just plain old regular cell division is going to divide that up into many individual cells. Okay? So here we have our lump of cells that's starting out life. Several weeks down the line, those cells start to differentiate and become specialized. They start to take on new tasks and new shapes, but we all just sort of start off as a lump of identical cells. Okay. Each one of these cells that we see in our beginning offspring, whatever species it is, are what we call stem cells. Those cells, the unique cells of the early embryo, have the potential to become any kind of cell. Okay, they're all the same. They are undifferentiated. They retain the ability to divide, and they can ultimately become all the different cell types of the body. So some of these stem cells are going to become nerve cells. Some of them are going to become muscle cells. Some are going to become blood cells. Some will go off to build bone. Others will become cartilage, right? These cells are going to ultimately change into different types of cells. But in the beginning, the original lump of cells, all of those cells have the potential to become any kind of cell. Okay? So when you hear people talk about stem cells, the reason stem cells are such a hot area of research for medical purposes is because they can become any kind of cell. So if we can trick that cell into becoming heart tissue, maybe we could grow a heart, right? If we could trick that cell into replacing a damaged cell in an organism's body, it could take on and replace something that's damaged, right? That's the reason that they're used so heavily in research. Once they start to specialize, they are undergoing a process known as differentiation. So differentiation is just the specialization of those early embryo cells. So they take on a particular structure and a particular function, and then they're dedicated to being that kind of cell. So once a cell becomes a muscle cell, that muscle cell will either never divide again in some cases, or it will only make more muscle cells. Right? Once a cell becomes a nerve cell, it's probably not going to divide again. So they're going to take on their sort of official task in the body as they go through differentiation. And some cells become permanently differentiated, which means they don't ever divide again after they've differentiated. So here, some of them take on a specific structure and function, and then they make new cells. Like your skin cells, all of your skin cells are coming from a basement layer of cells here that is constantly producing new cells, and then they push up to the surface, right? So those cells are dividing kind of all the time, making new cells, and you're just shedding them, shedding the outer surface as you go through life. Cells like neurons become permanently differentiated, and once they have that form, they actually don't ever divide again. And so the reason that it's so hard to recover from damage to neurons, nerve cells, is because we can't just grow a new one to replace or repair. They have to come from a stem cell. Well, there's an active growing period where those tissues are more able to regenerate than others. And there are some nerve cells that we thought couldn't regenerate, and now as neuroscience advances, we discover they can. And I haven't kept up on the exact mechanics of which cells can and don't. Obviously, as we're going through our growth and development period, our cells are more flexible than we get older. We regenerate faster, cell division occurs more readily, and the cells have more duplications left in them. As our cells age, we repair much slower, and we have much less ability to repair. And part of that comes from the fact that like the DNA actually changes in the cells over lifetime, and your cells are aging. So that's all kind of beyond this class, but there are complicated mechanisms that regulate that. Yes, your brain is quite large when you're born, but it does continue to grow. And actually, it's not fully developed, and the last neural connections aren't made in humans until you're in your early to mid 20s. So I think females, it's like 18 to 21, and males, it's like 21 to 25 or something like that before that frontal lobe finishes. Good time. It's why you shouldn't start lifelong habits in your early 20s. Like you should definitely avoid anything that could become an addiction in your late teens and early 20s, because if it happens while that closure is occurring in your brain, it's much more likely to become a lifelong habit versus a short-term habit. I think that's what the literature shows. Yeah, actually. And you're much less likely to start at 30. And if you, right, it's this like, yeah, right, right. And the pattern doesn't become as fixed. And there's also something about like language, your language learning center is super, super active, like in that like 4th, 5th, 6th, 7th grade time period. And it's the reason that you can remember all of the pop songs of your childhood, but you can't remember important things you're trying to learn in college. Because it's like those words were really a part of when your language center was developing. All sorts of crazy stuff in how that development occurs. Okay, so now we're going to actually move into your chapter, the stuff that's in your book. That's all my preamble, just to make sure we're on the same page. Okay, I feel like a lot of people don't really know very much about how cells combine, and so I just want to make sure we understand the difference between asexual and sexual reproduction and all of those things. As we move forward, we're going to take a really human-centric approach. Many of you are taking this as a prerequisite to a human-centric class. As I'm teaching Bio 2 right now, simultaneously to this class, I think a lot about all of the exceptions in the animal world. Like if you're a tapeworm, you can reproduce sexually or asexually. If you can't find a partner, you can line up all along the length of your body and sexually reproduce with yourself. And so like all of these things that you might not think about even being possible when you have a very human-centric view, crazy things happen in the animal world. Many animals start off one sex, switch to another sex, and then go back to their original sex. There's all sorts of crazy things that happen in the animal world. So this conversation, when we talk about sexual reproduction, we're going to have a very human-centric approach. But I want you to know that there's a great wide world of reproduction out there. It's fascinating. Okay. So the first thing that we need to talk about, first like term, unfortunately section 8.1 is really term heavy. I know you don't want a bunch of new terms coming at you before the exam, but we only got 15 more minutes. The first one is the genome. And when we talk about the term genome, we mean the full complement of DNA that a cell has. So every single one of the cells in my body has an identical genome, with the exception of any developing egg cells in my ovaries, right? All of the rest of my cells have all of the chromosomes that they're supposed to have, a full set, and they are identical, unless some mutation has happened along the way somewhere that I'm unaware of, okay? In eukaryotic organisms, which humans are one of, right? Everything except for the bacteria and the archaea. The genome is made up of many double-stranded linear DNA molecules. So we talked about the double helix. When we talked about DNA, we said it's 2 molecules that are twisted together like a rope ladder, right? Each one of these colored tubes in this diagram is, that's not the color I was going for, like this one here and this one here. Each one of those is a double-stranded linear DNA molecule. Okay. In the human body, we have 46 of these double-stranded linear DNA molecules. And in this picture, you might notice that they come in pairs. Here we've got the green pair. Over here we have a short blue pair. Here we have an even shorter, well actually, whoops. Let's see now. This one goes with this one, right? These two go together, these two go together. So we have all these pairs. So we have 46 of these double-stranded linear pieces of DNA, and they come in 23 types. We have 2 of each. So remember, half of the genetic material of the genome came from one parent, and the other half came from the other parent. So we got 23 pieces from the egg and 23 pieces from the sperm. We have 46 altogether. Does that make sense? So I'm going to keep using these numbers and talk in a very human-centric way because I think it helps us to keep track of what's happening. Okay. When we're looking at this particular picture, this circle here is the nucleus. So I'm really zoomed in on the cell. And this dark spot here is the nucleolus, which if you remember as you're reviewing for your next exam, is where ribosomes are synthesized. So when we look in a cell, we see this sort of dark region, the nucleolus, where the ribosomes are being synthesized. And then we just see this cloud, right? You don't see any defined structures. There's no nice pair of tubes like there was in the previous slide. And that's because in a cell that's just carrying out normal cell function, we have what we call chromatin. And that's the DNA complexed with proteins. And so we have individual proteins that are attached to the DNA molecules. And it's all just loose in there, kind of like a big pile of spaghetti. So I always sort of think of like the chromatin as just being a bunch of spaghetti noodles just kind of stretched out and laying inside the nucleus. Okay. This image we have of those individual pieces of DNA is when they're highly compacted and condensed and they're getting ready to travel. In normal cell function, they're just a big loose pile of DNA, and we call that chromatin. The DNA complex with its proteins, just at rest inside the nucleus. Let's see, we're skipping some slides here. Okay. When we get ready to do cell division, we can't just have this loose spaghetti pile of DNA if we want to copy it and organize it into 2 new cells, right? So I'm getting ready to go on a trip this weekend. When I'm ready to head to the train, I'm not going to walk to my closet, grab my 10 favorite outfits on their hangers, throw them over my arm, and head to the train, right? Because that would be a big pain in the butt to deal with. How am I going to put it somewhere? How am I going to carry it? What am I going to do when I get to Penn Station, right? I'm going to fold them up and put them in a suitcase and carry them in a little box, right? The cell does the same thing. Before we're going to move these chromosomes around, we got to package them up, make them small and manageable so that they're not going to get tangled up with each other, they're not going to get caught on each other and break, and they're not going to get disassociated. So this is when we actually start using those proteins to package the DNA up in preparation for mitosis, for cell division. Okay. So the proteins that were in the chromatin all along are known as histone proteins. And these histone proteins come in a little set. There's 1, 2, 3, 4. And on the underside, under here, there's 1, 2, 3, 4 more. So we have 8 little spherical proteins kind of together in a clump. And I don't know how many of you have any association with sewing or thread, but can you imagine like a plastic or wooden spool that thread is wound around like a bobbin? If you can visualize that, it's how I like to think of the proteins. Like we have this little unit, and we're going to wrap the DNA around it so that we can keep track of all of it and condense it and make it smaller. So instead of having a big, huge spaghetti pile, we wind it up on the histone proteins and compact it and make it smaller. So when the DNA is getting ready to be moved, it's going to package up. And so we're going to take our double helix here and loop it around the histone a few times. And then we have a little short run or linker DNA. And then we're going to wrap some around the next histone. And we have a little linker, and we're going to wrap it. And we continue to do that, forming these little bead-like units. Each one of these is known as a nucleosome. So a nucleosome is that histone protein bobbin with some DNA wrapped around it. Okay. And then we're just going to coil that string of bobbins up and further compact it down. So here's our double helix. It winds around the histone proteins to make little nucleosomes, and then we loop those nucleosomes back on themselves to make a thickened tube. And then just like folding up a garden hose, we loop that around and compact it more. So we can take this really, 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 really long piece of DNA and package up into this tiny little piece that's going to fit inside our nucleus and be compact and easy to move around. Okay. So what you see when you look at the images in this chapter of cells getting ready for cell division is you see these tube-shaped structures. And each tube-shaped structure is one double helix all compacted. This is actually a pair, one from each parent that are held together. Okay. So, unfortunately, for you guys, this is another one of those places where things were named at a time when we didn't really know what we were looking at. And then we discovered more, and we had to add more names and add more names. And there's some overlap in vocabulary here that I think gets really confusing for students. And so however you have to learn your vocabulary, whatever you have to do to try to keep these pieces separate, you're going to have to focus on trying to make sure that you understand the difference between these terms, because otherwise it can get pretty complicated. In order to divide the cell, the first thing we have to do is make a new copy of the DNA, because the DNA has all the instructions for everything the cell is ever going to do. So the new cell has to have a full set of instructions to function, right? So we're going to copy down the parent DNA and get it ready to give to the new offspring, okay? So the DNA has to be duplicated before division can start. When we were first talking about this, I've been referring to these tube-like structures, right? This is our DNA molecule, one double helix, and as we take that really, really long double helix and we wrap it around those nucleosomes and coil it up into a tube-like structure, we're going to represent it like this, and we call that a chromosome. So each one of those linear pieces of DNA inside the nucleus is a chromosome. Okay. When we get ready to do cell division, we have to make an identical copy. So just like if we could put our chromosome on the photocopier machine and make an exact match to it, the cell is going to copy down nucleotide for nucleotide an exact matching copy. Okay. Together, these two identical copies are sister chromatids. So each copy is a single chromatid, and together they are a pair of sister chromatids. So we took this, we made an identical copy, and now we have two identical copies of the same genetic material, and we attach them together by a little lasso of protein known as the centromere. Okay, so you have a lot of words that sound similar, and they all start with C. Everything starts with C. Chromatin, chromosome, chromatid, centromere. It's too much, right? So we have this pair of sister chromatids. They're two identical copies of the exact same information. They really only carry one chromosome's worth of information. So we also call them a chromosome, even though it's two items that are together, because they're only representing one piece of genetic information. Okay. So the sister chromatids are the two identical copies of the same chromosome, and the centromere is the protein that attaches them together and holds them together. Later, when we talk about cell division, the microtubules that are essential for cell division are going to attach at the centromere region. Okay. So, what does it mean to be a chromosome? A chromosome is an individual double helix strand of DNA, right? So even when they're in their loose spaghetti configuration, each linear strand is a chromosome. It is still a chromosome when it packages up into its condensed tube form ready to travel. A pair of sister chromatids is still a single chromosome because it represents one piece of the genetic information, right? Each cell has 46 chromosomes in a human body, 46 pieces of genetic information. Just because we have two copies of each of them doesn't mean that we have more information or more instructions. We just have more copies of the same information. So it's still only 46 chromosomes. The word chromosome, some means body, chromo means colored. It's a colored body. When we first discovered these in really early microscopy, playing around with stains for cells, somebody looked through a microscope and was like, oh, look, there's a colored structure. I'll call it a chromosome. That's where the name comes from. And since then, as it came to mean something more than just a blob of color in our stain, we had to sort of expand our definition. So that's why a chromosome is kind of a diverse suite of things. It's either a single linear piece of DNA in its loose form, in its compacted form for travel, or a pair of sister chromatids duplicated and ready for cell division. So we'll talk about that a little bit. There are a number of conditions where individuals can either have extra chromosomes or missing chromosomes. Generally, if you're missing one chromosome or more, you don't develop into a viable offspring. So an embryo that is missing chromosomes, if it's missing all but one of those chromosomes, it's probably not going to make it to birth. If it makes it to live birth, it's not going to live through its juvenile years, generally speaking. There is an exception to that, which is if you're missing a sex chromosome, you can be all right. And so you may not be fertile, you may have some other issues, but if you're missing a sex chromosome, it's okay. So we'll talk about some of those exceptions later. It's more common to have extra chromosomes than missing chromosomes because that absence of information causes malfunction in the developing embryo. Okay, so this is just another way to look at this. And in this diagram, I want to add in one other thing, which is that in your original nucleus, before we start to duplicate things for movement, we have our 46 chromosomes, but they only come in 23 types, right? 23 of them came from one parent, and the other 23 came from the other parent. Okay. It is, except for in the sex chromosome. So there's some organisms that get one fewer chromosome. If they're like a male has one fewer chromosome than a female, or vice versa. And so in this case, we have half of our chromosomes came from the sperm, half of our chromosomes came from the egg. They're a matched set though. They're like salt and pepper shakers. They're similar, but not quite identical. They carry the same information, but it's not the exact same information. And so we call that pair a homologous pair. And so these two together, these two copies of chromosome number 1, if that's its ID, are a homologous pair of chromosomes. When they duplicate, they become a pair of sister chromatids, but they still have a mate out there in the nucleus that is the other half of the homologous pair. Okay. At what point? Okay, so in this chapter, in cell division, the maternal and the paternal chromosome are just free floating in the nucleus and they don't actually come together. When we prepare for sexual reproduction, they will actually temporarily line up together. That only happens in the ovaries and the testes. It doesn't happen anywhere else in the body. Okay. All right, so we'll stop here. We'll do a quick review of what is a chromosome when we come back after our exam and move forward with this chapter. Okay, you've got a while to get these terms straight. Don't think about them for another week because you're going to be preparing for your exam, right?